Alright guys, welcome back to Underground Science, and in this video we're going to be talking about pyrimidine synthesis, okay? So it's important to note that, what the pyrimidines even are. So we have thymine, cytosine, uracil. Uracil is only an RNA, okay? So first things first, we need carbamoyl phosphate, okay? So the way we're going to do that, we're going to take glutamine, CO2, ATP, and make carbamoyl phosphate. But in order to synthesize carbamoyl phosphate, I need a synthetase, specifically a carbamoyl phosphate synthetase, type 2. Because remember, type 1's in the urea cycle, which we'll talk about later, okay, in another video. So carbamoyl phosphate synthetase type 2 is needed to form carbamoyl phosphate. Because it's a synthetase, we need ATP, okay? Now I want to go from carbamoyl phosphate to carbamoyl aspartate. So I do that via aspartate carbamoyl transferase. Okay, again, from carbamoyl aspartate, the goal is to go to dihydroorotate. Okay, but I can only do that if I have the enzyme dihydroorotase. Okay, and we dehydrate this carbamoyl aspartate by taking out a water molecule. Okay, so now with the help of dihydroorotase, I'm at dihydroorotate. Now we go from dihydroorotate to orotic acid. Okay, or orotate. So again, dihydroorotate to orotic acid. And I do that via dihydroorotate dehydrogenase. And this enzyme, fun fact, is found in the mitochondria. Okay? So we went from dihydroorotate to orotic acid, okay, via dihydroorotate dehydrogenase. Now that we have orotic acid, we can combine orotic acid with a molecule that's called phosphoribosyl pyrophosphate. Okay, and this PRPP molecule is important because it's going to donate its ribose phosphate group to orotic acid. Okay, and we'll talk about this more when we talk about purine synthesis. But for now, know that we use a molecule called phosphoribosyl pyrophosphate. This will donate its ribose phosphate group to orotic acid so we can form oritidine 5' monophosphate, meaning this phosphate that we have is on the 5 carbon. Okay, on the 5th carbon. So we go from orotic acid to oritidine monophosphate, or OMP, and we do this via orotate phosphoribosyl transferase. Okay, so that's the enzyme right here. Orotate phosphoribosyl transferase. Now we're at OMP. All right, cool. So let's zoom out just a tad bit and then scroll down. Okay, so we're at OMP now, right? Now we have to go from OMP to UMP. So the way we're going to do that is we're going to decarboxylate OMP with the help of an enzyme called OMP decarboxylase. All right, so again, from orotic acid, we went from orotic acid to OMP. Okay, so the way we did that was we used the help of a PRPP molecule and orotate phosphoribosyl transferase. Okay, so now that we have OMP, we go from OMP to UMP via OMP decarboxylase. So the fun thing about this is, right, or a cool thing about this is that, so remember, orotate phosphoribosyl transferase went from orotic acid to OMP, right? And this enzyme, OMP decarboxylase, which helped us go from OMP to UMP, they both form a dimer protein, which is UMP synthase. Okay, so UMP synthase is made up of this enzyme and this enzyme. So that means UMP synthase helps make UMP, okay, which we already formed right here, uridine monophosphate. Now that we have uridine monophosphate, we just add a phosphate to it and get UDP, all right? So after we form UDP, okay, from UMP, this can go two ways, because remember, this is pyrimidine synthesis. So we already formed what we need for uracil, right? We formed UMP, so that's relating to RNA, right? Uridine monophosphate can go participate in RNA synthesis. Now we need to form cytosine and thymine, because those are the other two pyrimidines. So UDP can now get converted into deoxy-UDP, okay? But see how that converts from RNA to DNA? So you need an enzyme to do that, and that's ribonucleotide reductase. Okay? So now that you have DUDP, you can convert that into deoxy-UMP, and then now you can take this and form deoxy-TMP. So what's this going to be used for? DNA, right? Deoxythymidine monophosphate. So the way we convert deoxyuridine monophosphate to deoxythymidine monophosphate is via thymidylate synthase, okay? But something else has to happen here. So we take 5,10-methylene tetrahydrofolate, and that converts to dihydrofolate, okay? Dihydrofolate. So again, 5,10-methylene tetrahydrofolate converts to dihydrofolate. Then dihydrofolate is converted to tetrahydrofolate via the help of an enzyme called dihydrofolate reductase. Then tetrahydrofolate gets converted into 5,10-methylene tetrahydrofolate. That again participates in this reaction, gets converted back to dihydrofolate, and then this is a whole cycle. Okay, so dihydrofolate reductase is an important enzyme in this step when we convert deoxyuridine monophosphate to deoxythymidine monophosphate. Okay, and we also need thymidylate synthase for this. So now that we have deoxythymidine monophosphate, we have deoxyuridine monophosphate, right? Or specifically uridine monophosphate right here. Okay, we have both of those. Now let's try to see how we can start working with cytidine and cytosine, right? So from UDP, remember, this is one path that we took. Another path to take is to form CTP, or cytidine triphosphate. The way we're going to do that is notice how in UDP, we only have diphosphate. So we only have two phosphates here, but this is CTP, or cytidine triphosphate. That means you added another phosphate. And the way we did that was with the help of ATP. So we use an ATP here, all right, and we formed cytidine triphosphate via the help of CTP synthetase. Remember synthetase, you would use an ATP. And we also use an amine group from glutamine, okay? So now that we have CTP, we can just convert that, or basically we can just strip off a phosphate and form CDP, or cytidine diphosphate. And then remember, when we go from something that's, from RNA to DNA, right, because deoxy versus no deoxy, we use the enzyme ribonucleotide reductase. So we use that, and we get deoxycytidine diphosphate. This can get converted to deoxycytidine monophosphate, and then that makes our three main pyrimidines, right? Because we have our cytidine, we have our uridine, and we have our thymidine, right? So uridine right here, and then we have our thymidine all the way down here. So that's basically it. Those are the three pyrimidines that we just made. And in the future videos, what we can talk about is now the drugs that work on these enzymes in pyrimidine synthesis, all right? And so we're going to talk about that in the next few videos. And that's basically it for this video, and I hope to see you guys in the next videos.